Chapter 3 Plant Kingdom. Algae, bryophytes, pteridophytes, gymnosperms, angiosperms, plant life cycles and ordination of generations. In the previous chapter, we looked at the broad classification of living organisms under the system proposed by Whittaker in 1969, wherein he suggested the five-kingdom classification was Monera, Protista, fungi, Animalia, and Plantae. In this chapter, we will deal in detail with further classification within kingdom Plantae, popularly known as plant kingdom. We must stress here that our understanding of the plant kingdom has changed over time. Fungi and members of the Monera and Protista having cell walls have now been excluded from Plantae, though earlier classifications placed them in the same kingdom. So, the cyanobacteria that are also referred to as blue-green algae are not algae anymore. In this chapter, we will describe algae, bryophytes, pteridophytes, gymnosperms, and angiosperms under Plantae. Let us also look at the classification within angiosperms to understand some of the concerns that influence the classification systems. The earlier systems of classification used only gross superficial morphological characters such as habit, color, number, and shape of leaves, etc. They were based mainly on vegetative characters or on the androecium structure system given by Linnaeus. Such systems were artificial. They separated the closely related species since they were based on a few characteristics. Also, the artificial systems gave equal weightage to vegetative and sexual characteristics. This is not acceptable since we know that often the vegetative characters are more easily affected by environment. As against this, natural classification systems developed which are based on natural affinities among the organisms and consider not only the external features but also internal features like ultrastructure, anatomy, embryology, and phytochemistry. Such a classification for flowering plants was given by George Bentham and Joseph Dalton Hooker, which were not only the external features but also internal features like ultrastructure, anatomy, embryology, and phytochemistry, given by George Bentham and Joseph Dalton Hooker. At present, phylogenetic classification systems based on evolutionary relationships between the various organisms are acceptable. This assumes that organisms belonging to the same taxa have a common ancestor. We now use information from many other sources too to help resolve difficulties in classification. These become more important when there is no supporting fossil evidence. Numerical taxonomy, which is now easily carried out using computers, is based on all observable characteristics. Number and codes are assigned to all the characters and the data are then processed. In this way, each character is given equal importance and at the same time hundreds of characters can be considered. Cytotaxonomy, that is based on cytological information like terms of number, structure, behavior, and chemotaxonomy that uses the chemical constituents of the plant to resolve confusions are also used by taxonomists these days. Algae are chlorophyll bearing, simple, thalloid, autotrophic, and largely aquatic, both freshwater and marine organisms. They occur in a variety of other habitats, moist stones, soils, and wood. Some of them also occur in association with fungi, lichen, and animals, for example, on sloth bear. The form and size of algae is highly variable, ranging from colonial forms like volvox and the filamentous forms like Ulothrix and Spirogyra, figure 3.1. A few of the marine forms, such as kelps, form massive plant bodies. The algae reproduce by vegetative, asexual, and sexual methods. Vegetative reproduction is by fragmentation. Each fragment develops into a thallus. Asexual reproduction is by the reproduction of different types of spores, the most common being the zoospores. They are flagellated, motile, and germination gives rise to new plants. Sexual reproduction takes place through fusion of two gametes. These gametes can be flagellated and similar in size as in Ulothrix, or non-flagellated, non-motile, but similar in size as in Spirogyra. Such reproduction is called isogamous. Fusion of two gametes dissimilar in size as in species of Eudorina is termed as anisogamous. Fusion between one large non-motile static female gamete and a smaller motile male gamete is termed oogamous, for example, volvox fucus. Figure 3.1 showing green algae, A1 volvox, 2 Ulothrix. Volvox is colonial, daughter colony, and this is the parent colony. B brown algae, 1 showing laminaria, 2 fucus. Laminaria has got front, stipe, and holdfast. Leaf-like front, stipe, and holdfast. 2 Fucus has got air bladder, front and midrib, and a holdfast. B3, Dictyota, having front and stipe. So, green algae wallbox, it is colonial with parent colony and daughter colony. 2, Eulothrix. Brown algae, laminaria, having front, stipe and holdfast. Fucus having air bladder, front, midrib, holdfast, and Dictyota with front, stipe. C. Red algae, this is Porphyra. 1. Porphyra with front. 2. Polysiphonia with main axis and the branches. Algae are useful to man in a variety of ways. At least a half of the total carbon dioxide fixation on Earth is carried out by algae through photosynthesis. Being photosynthetic, they increase the level of dissolved oxygen in their immediate environment. They are of paramount importance as primary producers of energy-rich compounds which form the basis of food cycles of all aquatic animals. Many species of Porphyra, Laminaria and Sargassum are among the 70 species of marine algae used as food. Certain marine brown and red algae produce large amounts of hydrocolloids, water-holding substances, for example algin, brown algae and carrageen red algae, which are used commercially. Agar, one of the commercial products obtained from Gelidium and Gracilaria, are used to grow microbes and in preparations of ice creams and jellies. Chlorella, a unicellular alga rich in proteins, is used as food supplements even by space travelers. The algae are divided into 3 main classes: Chlorophyceae, Phaeophyceae and Rhodophyceae. Chlorophyceae. The members of Chlorophyceae are commonly called green algae. The plant body may be unicellular, colonial or filamentous. They are usually grass green due to the dominance of pigments chlorophyll A and B. The pigments are localized in definite chloroplasts. The chloroplasts may be discoid, plate-like, reticulate, cup-shaped, spiral or ribbon-shaped in different species. Most of the members have one or more storage bodies called pyrenoids located in the chloroplast. Pyrenoids contain protein besides starch. Some algae may store food in the form of oil droplets. Green algae usually have a rigid cell wall made of an inner layer of cellulose and an outer layer of pectose. Green algae have a rigid cell wall made of an inner layer of cellulose and an outer layer of pectose. Vegetative reproduction usually takes place by fragmentation or by formation of different types of spores. Asexual reproduction is by flagellated zoospores produced in zoosporangia. The sexual reproduction shows considerable variation in the type and formation of sex cells, and it may be isogamous, anisogamous or oogamous. Some commonly found green algae are Chlamydomonas, Volvox, Eulothrix, Spirogyra and Cara. Figure 3.1a. Phaeophyceae. Members of Phaeophyceae brown algae are found primarily in marine habitats. They show great variation in size and form. They range from simple branched filamentous forms, ectocarpus, to profusely branched forms as represented by kelps, which may reach a height of 100 meters. They possess chlorophyll A, C, carotenoids and xanthophylls. They vary in color from olive green to various shades of brown depending upon the amount of xanthophyll pigment fucosanthin present in them. Food is stored as complex carbohydrates which may be in the form of laminarin or mannitol. The vegetative cells have a cellulosic wall usually covered on the outside by a gelatinous coating of algin. The protoplast contains, in addition to plastids, a centrally located vacuole and nucleus. The plant body is usually attached to substratum by a holdfast and has a stalk, the stipe, and a leaf-like photosynthetic organ, the frond. The vegetative cells have a cellulosic wall usually covered on the outside by a gelatinous coating of algin. Algin is a gelatinous coating. Food is stored as complex carbohydrates in the form of laminarin or mannitol in Phaeophyceae, brown algae. The vegetative cells have a cellulosic wall usually covered on the outside by a gelatinous coating of algin. The protoplast contains, in addition to plastids, a centrally located vacuole and nucleus. The plant body is usually attached to substratum by a holdfast. The plant body is usually attached to substratum by a holdfast and has a stalk, the stipe. The stalk is called the stipe and leaf-like photosynthetic organ in the front. Vegetative reproduction takes place by fragmentation. Asexual reproduction in most brown algae is by biflagellate zoospores. Biflagellate zoospores are the asexual reproduction. Takes place by Vegetative reproduction takes place by fragmentation. Asexual reproduction in most brown algae is by biflagellate zoospores that are pear-shaped and have two unequal laterally attached flagella. Unequal laterally attached flagella, two of them, and they are pear-shaped. Biflagellate zoospores are pear-shaped and have two unequal laterally attached flagella. Sexual reproduction may be isogamous and isogamous or oogamous. Union of gametes may take place in water or within the oogonium, oogamous species. For the oogamous species, union of gametes takes place within the oogonium or they take place in water. The gametes are pyriform, pear-shaped and bear two laterally attached flagella. The common forms of Ectocarpus dictyota, laminarum, sargassum and fucus. Common forms are Ectocarpus, dictyota, laminaria, sargassum and fucus. Rhodophyceae. Members of Rhodophyceae are commonly called red algae because of the predominance of red pigments or phycoerythrin in their body. Majority of the red algae are marine with greater concentration found in the warmer areas. They occur in both well-lighted regions close to the surface of water and also at great depths in oceans where relatively little light penetrates. The red thalli of most of the red algae are multicellular. Some of them have complex body organization. The food is stored as Floridian starch, which is very similar to amylopectin and glycogen in structure. The red algae usually reproduce vegetatively by fragmentation. They reproduce asexually by non-motile spores and sexually by non-motile gametes. Sexual reproduction is oogamous and accompanied by complex post-fertilization developments. The common members are Polycyphonia, Porphyra, Gracilaria and Gelidia. Rhodophyceae, let's read that again. The members of Rhodophyceae are commonly called red algae because of the predominance of red pigments or phycoerythrin in their body. Majority of the red algae are marine with greater concentration found in the warmer areas. They occur in both well-lighted regions close to the surface of water and also at great depths in ocean where relatively little light penetrates. The red thalli of most of the red algae are multicellular. Some of them have complex body organization. The food is stored as Floridian starch, which is very similar to amylopectin and glycogen in structure. Divisions of algae and their main characteristics. Classes, common name, major pigments, stored food in them, cell wall, flagellar number and position of insertions, habitat. Chlorophyceae, green algae with chlorophyll A, B. Stored food is starch, cell wall is of cellulose, flagellar number and position of insertions 2 to 8 flagella, equal and apical. Habitat, freshwater, brackish water and saltwater. Brackish water, saltwater. Phaeophyceae, brown algae, chlorophyll A, C, fucoxanthin. Stored food is mannitol, laminarin. Cell wall is cellulose and algin. Flagella 2, unequal and lateral. Freshwater, rare, brackish water, saltwater. Rhodophyceae, red algae, chlorophyll A, D, phycoerythrin. Floridian starch is the stored food. Cell wall of cellulose, pectin and polysulfate esters. Flagella is absent. Habitat, freshwater, some brackish water, saltwater, most. The red algae usually reproduce vegetatively by fragmentation. They reproduce asexually by non-motile spores and sexually by non-motile gametes. Sexual reproduction is by oogamous and accompanied by complex post-fertilization developments. The common members are Polycyphonia, Porphyra, Gracilaria and Gelidium. Bryophytes. Bryophytes include the various mossel and liverworts that are found commonly growing in moist shaded areas in the hills. 3.2 Bryophytes. A liverwort Markensia. A is the female Thalassomachensia with archegoniophore gemicup. gemmicub, and rhizoids. These are the rhizoids. This is the gemmicub, and female thallus of Mackentia, a liverwort, has archegoniophore, and male thallus have antheridophore. It also has gemmicub and rhizoids. Moses, C. funaria, is a gametophyte and sporophyte sphagnum. Gametophyte and sporophyte of funaria, and D. sphagnum, this is gametophyte of sphagnum. So funaria's gametophyte has main axis and rhizoids. This is funaria, has main axis and rhizoids, and the sporophyte main axis. Gametophyte has main axis, rhizoids, and leaves. Sporophytes have setae and capsule and setae in sporophyte, and main axis and rhizoids and leaves present in gametophyte of funaria. D is sphagnum gametophyte. This is gametophyte with archegonial branch, antheridial branch. These are the other branches. Bryophytes are also called amphibians of plant kingdom because of these. Plants can live in soil but are dependent on water for sexual reproduction. Bryophytes are also called amphibians of plant kingdom because these plants can live in soil but are dependent on water for sexual reproduction. They are dependent on water for sexual reproduction. So they are called the amphibians of plant kingdom, though they live in soil. They usually occur in damp, humid and shaded localities. They play an important role in plant succession on bare rocks or soil. The plant body of bryophytes is more differentiated than that of algae. It is thallus-like and prostrate or erect. The plant body of bryophytes is more differentiated than that of algae. It is plant body of bryophytes is more differentiated than algae. It is thallus-like and prostrate or erect and attached to the substratum by unicellular multicellular rhizoids. They lack true roots, stem or leaves. They may possess root-like, leaf-like or stem-like structures. The main plant body of the bryophyte is haploid. It produces gametes, hence it is called gametophyte. Gametophyte is haploid. It is the main plant body of bryophyte is gametophyte, and it produces gametes. The sex organs in bryophytes are multicellular The sex organs in bryophytes are multicellular. The male sex organ is called antheridium. They produce biflagellate anthrozoids. The female sex organ is called archegonium, is flask-shaped and produces a single egg. So the male sex organ is called antheridium. They produce biflagellate anthrozoids. The female sex organ called archegonium is flask-shaped and produces a single egg. The anthrozoids are released into water where they come in contact with archegonium. And they come in close contact with archegonium. Archegonium is flask-shaped and produces a single egg. An anthrozoid fuses with the egg to produce the zygote. Zygotes do not undergo reduction division. They do not undergo reduction division immediately. They produce a multicellular body called sporophyte. The sporophyte is not free living but attached to this photosynthetic gametophyte and derives nourishment from it. Some cells of the sporophyte undergo reduction division, meiosis, to produce a haploid spore. Some cells of sporophytes undergo reduction division to produce haploid spores. These spores germinate to produce gametophyte. Bryophytes in general are of little economic importance, but some mosses provide food for herbaceous mammals, birds, and other animals. Bryophytes in general are of little economic importance, but some mosses provide food for herbaceous mammals, birds, and other animals. Species of sphagnum, a moss, provide peat that have long been used as fuel. Peat is used as species of sphagnum, a moss. Sphagnum is a moss which provides peat, which is used as a fuel. And is packing material for transshipment of living material because of their capacity to hold water. Most among with lichens are the first organisms to colonize rocks and hence are of great ecological importance. They decompose the rocks, making the substrate suitable for the growth of higher plants. Since mosses form dense mats on the soil, they reduce the impact of falling rain and prevent soil erosion. Since mosses form dense mats on the soil, they reduce the impact of falling rain and prevent soil erosion. They reduce the impact of falling rain and prevent soil erosion. The bryophytes are divided into liverworts and mosses. Liverworts. The liverworts grow usually in moist, shady habitats such as banks of streams, marshy ground, damp soil, bark of trees, and deep in the woods. The plant body of a liverwort is thalloid, for example, Markinsia. The thallus is dorsiventral and closely oppressed to the substrate. The thallus is dorsiventral and closely oppressed to the substrate. The leafy members have tiny leaf-like appendages in two rows on the stem-like structures. The leafy members have tiny leaf-like appendages in two rows on the stem-like structures. The leafy members have tiny leaf-like appendages in two rows on the stem-like structures. Asexual reproduction in liverworts takes place by fragmentation of thalli or by the formation of specialized structures called gemmae. Singular gemma. Gemmae are green, multicellular, asexual buds which develop in small receptacles called gemmicups located on the thalli. The gemmae become detached from the parent body and germinate to form new individuals. During sexual reproduction, male and female sex organs are produced either on the same or on different thalli. The sporophyte is differentiated into a food, seta, and capsule. After meiosis, spores are produced within the capsule. These spores germinate to form free-living gametophytes. Organs are produced either on the sex organs are produced either on the same or on different thalli. The gemmae become detached from the parent body and germinate to form new individuals. During sexual reproduction, male and female sex organs are produced either on the same or on different thalli. The sporophyte is differentiated into food, seta, and capsule. After meiosis, spores are produced within the capsule. These spores germinate to form free-living gametophytes. Mosses. The predominant stage of the life cycle of moss is the gametophyte, which consists of two stages. The first stage is the protonema stage, which develops directly from a spore. It is a creeping, green, branched, and frequently filamentous stage. The second stage is the leafy stage, which develops from the secondary protonema as a lateral bud. They consist of upright, slender axis bearing spirally arranged leaves. They are attached to the soil through multicellular and branched rhizoids. This stage bears the sex organs. Vegetative reproduction in mosses is by fragmentation and budding in the secondary protonema. In sexual reproduction, the sex organs antheridia and archegonia are produced at the apex of the leafy shoots. After fertilization, the zygote develops into a sporophyte consisting of food, seta, and capsule. The sporophyte in mosses is more elaborate than that in liverworts. The capsule contains spores. Spores are formed after meiosis. The mosses have an elaborate mechanism of spore dispersal. Common examples of mosses are Funaria, Polytrichum, and Sphagnum. The common examples of mosses are Funaria, Polytrichum, and Sphagnum. The mosses have an elaborate mechanism of spore dispersal. Common examples of mosses are Funaria, Polytrichum, and Sphagnum. Funaria, Polytrichum, and Sphagnum. Pteridophytes. The pteridophytes include horsetails and ferns. Pteridophytes are used for medicinal purposes and as soil binders. They are used for medicinal purposes and as soil binders. They are also frequently grown as ornamentals. Evolutionarily, they are the first terrestrial plants to possess vascular tissue, xylem and phloem. We shall study more about these tissues in Chapter 6. The pteridophytes are found in cool, damp, and shady places, though some may flourish well in sandy soil conditions. You may recall that in bryophytes, the dominant phase in the life cycle is the gametophytic plant body. However, in pteridophytes, the main plant body is a sporophyte, which is differentiated into two root stem and leaves. These organs possess well-differentiated vascular tissues. The leaves in pteridophyta are small microphyls, as in Selaginella, or large macrophyls, as in ferns. The sporophytes bear sporangia that are subtended by leaf-like appendages called sporophylls. These organs possess well-differentiated vascular tissues. The leaves in pteridophyta are small microphyls, as in Selaginella, or large macrophyls, as in ferns. Small microphyls, as in Selaginella, or large macrophyls, as in ferns. The sporophytes bear sporangia that are subtended by leaf-like appendages called sporophylls. The sporophytes bear sporangia that are subtended by leaf-like appendages called sporophylls. In some cases, sporophylls may form distinct compact structures called strobili or cones. In some cases, sporophylls may form distinct compact structures called strobili or cones. Selaginella equisitum. The sporangia produce spores by meiosis in spore mother cells. The spores germinate to give rise to inconspicuous, small but multicellular, free-living, mostly photosynthetic thalloid gametophytes called prothallus. These gametophytes require cool, damp, and shady places to grow. Because of this specific restricted requirement and the need for water for fertilization, the spread of living pteridophytes is limited and restricted to narrow geographical regions. The gametophytes bear male and female sex organs called antheridia and archegonia respectively. Water is required for transfer of anthrozoids, the male gametes released from the antheridia, to the mouth of archegonium. Water is needed for transfer. From the antheridia to the mouth of archegonium. Fusion of male gamete with the egg present in the archegonium results in the formation of zygote. The zygote thereafter produces a multicellular, well-differentiated sporophyte, which is dominant phase of pteridophyte. The dominant phase of pteridophyte is sporophyte stage. Sporophyte stage is the dominant phase of pteridophytes. In majority of the pteridophytes, all the spores are of similar kinds. In majority of pteridophytes, all spores are of similar kinds. Such plants are called homosporous. Genera like Selaginella and Salvinia, which produce two kinds of spores, macro large and micro small spores, are known as heterosporous. The megaspores and microspores germinate and give rise to female and male gametophyte respectively. The female gametophyte in these plants are retained on the parent sporophyte for variable periods. The development of the zygote into young embryo takes place within the female gametophytes. This event is a precursor of the seed habits considered an important step in evolution. The pteridophytes are further classified into four classes: Coleopsida coleotum, Lycopsida, Selaginella lycopodium, Sphenopsida equisitum, and Pteropsida, Dryopteris, Teres, Adianthum. Pteridophytes: A. Selaginella with leaves, stem, roots. This is Selaginella. This is a pteridophyte. Selaginella with leaves, stem, roots. B is equisitum with strobilus, node, internode, branch, rhizome. This is equisitum. Equisitum with strobilus, node, internode, branch, and rhizome. C is a fern, and D is Salvinia. Gymnosperms. The gymnosperms, gymnosnaked spermacetes, are plants in which the ovules are not enclosed by any ovary wall and remain exposed both before and after fertilization. The seeds that develop post-fertilization are not covered, that is, are naked. Gymnosperms include medium-sized trees or tall trees and shrubs. Figure 3.4. Gymnosperms: A. Cycas, B. Pinus, C. Ginkgo, with a dwarf shoot, long shoot and seeds. One of the gymnosperms, the giant redwood tree sequoia, is one of the tallest tree species. The roots are generally tap roots. Roots in some genera have fungal association in the form of mycorrhiza pinus, while in some others, cycas, small specialized roots called coralloid roots are associated with nitrogen fixing cyanobacteria. The stems are unbranched cycas or branched pinus cedrus. The leaves may be simple or compound. In cycas, the pinnate leaves persist for a few years. The The leaves in gymnosperms are well adapted to withstand extremes of temperature, humidity, and wind. In conifers, the needle-like leaves reduce the surface area. Their thick cuticle and sunken stomata also help to reduce water loss. The gymnosperms are heterosporous. They produce haploid microspores and megaspores. The two kinds of spores are produced within the sporangia that are borne on sporophylls, which are arranged spirally along an axis to form lax or compact strobili or cones. The strobili bearing microsporophylls and microsporangia are called microsporangiate or male strobili. The microspores develop into a male gametophytic generation, which is highly reduced and is confined to only a limited number of cells. This reduced gametophyte is called a pollen grain. The development of pollen grains takes place within the microsporangia. The cones bearing megasporophylls with ovules or microsporangia are called macrosporangiate or female strobili. The cones bearing megasporophylls with ovules or megasporangia are called macrosporangiate or female strobili. The male or female cones of strobili may be born on the same tree, pinus. However, in cycas, male cones and megasporophylls are born on different trees. The megaspore mother cell is differentiated from one of the cells of the nucellus. The nucellus is protected by envelopes and the composite structure is called an ovule. The nucellus is protected by envelopes and the composite structure is called an ovule. The ovules are borne on megasporophylls, which may be clustered to form the female cones. The ovules are borne on megasporophylls, which may be clustered to form the female cones. The megaspore mother cell divides meiotically to form 4 megaspores. The megaspore mother cell divides meiotically to form 4 megaspores. One of the megaspore enclosed within the megasporangium develops into a multicellular female gametophyte that bears 2 or more archegonia or female sex organs. The multicellular female gametophyte is also retained within megasporangium. The multicellular female gametophyte is also retained within megasporangium. Unlike bryophytes and pteridophytes, in gymnosperms, the male and female gametophyte do not have an independent free-living existence. Unlike bryophytes and pteridophytes, in gymnosperms, male and female gametophyte do not have an independent free-living existence. They remain within the sporangia, retained on the sporophytes. The pollen grain is released from the microsporangium. They are carried in air currents and come in contact with the opening of the ovules borne on megasporophylls. The pollen tube carrying the male gametes grows towards archegonia in the ovules and discharges their contents near the mouth of archegonia. Following fertilization, zygote develops into an embryo and the ovules into seeds. These seeds are not covered. Gymnosperms, cycas, pinus, and ginkgo. Angiosperms. Angiosperms. Unlike the gymnosperms, where the ovules are naked, in the angiosperms or flowering plants, the pollen grains and ovules are developed in specialized structures called flowers. In angiosperms, the seeds are enclosed in fruit. The angiosperms are an exceptionally large group of plants occurring in a wide range of habitats. They range in size from the smallest wolfia to tall trees of eucalyptus over 100 meters. They provide us with food, fodder, fuel, medicines, and several other commercially important products. They are divided into two classes, the dicotyledonous and the monocotyledonous. The dicotyledons and monocotyledons. Figure 3.5a, angiosperms A showing a dicotyledon and B showing a monocotyledon. The dicotyledons are characterized by seeds having two cotyledons, reticulate venation. Reticulate venations in leaves and tetramerous or pentamerous flowers, that is having 4 or 5 members in each floral whorl. Monocotyledons, on the other hand, are characterized by single cotyledonous seeds, parallel venation in leaves, and trimerous flowers, having 3 members in each floral whorls. The male sex organ in a flower is a stamen. Each stamen consists of a slender filament with an anther at the tip. Within the anthers, the pollen mother cell divides by meiosis to produce microspores which matures into pollen grains. The female sex organ in a flower is a pistil. Pistil consists of a swollen ovary at its base. A long slender style and stigma consists of a swollen and stigma. Pistil consists of a swollen ovary at its base, a long slender style, and stigma. Inside the ovary, ovules are present. Generally, each ovule has a megaspore mother cell and that undergoes meiosis to form 4 haploid megaspores. 3 of them degenerate and one divide to form the embryo sac. Each embryo sac has a 3-celled egg apparatus, 1 egg cell and 2 synergids, 3 antipodal cells and 2 polar nuclei. The polar nuclei eventually fuse to produce a diploid diploid secondary nucleus. Pollen grain, after dispersal from the anthers, are carried by wind or various other agencies to the stigma of a pistil. This is termed as pollination. The pollen grains germinate on the stigma and the resulting pollen tubes grow through the tissues of stigma and style and reach the ovule. The pollen tubes enter the embryo sac where 2 male gametes are discharged. One of the male gametes fuses with the egg cell syngamy to form zygote. The other male gamete fuses with the diploid secondary nucleus to produce the triploid primary endosperm nucleus, PEN. Because of the occurrence of 2 fusions, that is syngamy and triple fusion, this event is termed as double fertilization and even unique to angiosperms. The zygote develops into an embryo with 1 or 2 cotyledons, and the PEN develops into endosperm, which produces nourishment to the developing embryo. The synergids and antipodals degenerate after fertilization. During these events, the ovules develop into seed and the ovaries develop into fruit. The life cycle of an angiosperm is seen in figure 3.6. Life cycle of an angiosperm. Sporophyte, stigma, and the sporophyte grows into anther and filament containing stamen, which has microsporidium, which produces microspores. This is a functional microspore or pollen grain. This is the microspore pollen grain with the vegetative and generative nucleus inside them, which grows into male gametophyte. Pollen grain is a male gametophyte. Pollen tube grows from it and enters through the stigma and style to the egg apparatus embryo sac. This is the egg apparatus. In the male gametes through synergids get into the egg apparatus, and zygote divides and forms embryo, and embryos divide to form the sporophyte. So here sporophyte grows into stamen. Sporophyte has stamen and pistil with stigma, style and ovary. This grows into the megaspore mother cell. This is the megasporangium ovule. This is the functional megaspore in the embryo sac. So here, this much is the gametophytic N generation, and this is sporophytic or 2N generation, life cycle of angiosperm. Plant life cycles and ordination generations. In plants, both haploid and diploid cells can divide by mitosis. This ability leads to the formation of different plant bodies, haploid and diploid. The haploid plant body produces gametes by mitosis. This plant body represents a gametophyte. Following fertilization, the zygote also divides by mitosis to produce the diploid sporophytic plant body. Haploid spores are produced by this plant body by meiosis. These in turn divide by mitosis to form a haploid plant body once again. Thus, during the life cycle of any sexually reproducing plant, there is an ordination generation between gamete producing haploid gametophyte and spore producing diploid sporophyte. However, different plant groups as well as an individual representing them differ in following patterns. 1. Sporophytic generation is represented only by the one-celled zygote. There are no free-living sporophytes. Meiosis in the zygote results in the formation of haploid spores. The haploid spores divide mitotically and forms the gametophyte. The dominant photosynthetic phase in such plants is the free-living gametophyte. This kind of life cycle is termed as haplontic. Many algae such as volvox, paragera and some species of Chlamydomonas represent this pattern. Figure 3.7a. Life cycle patterns of haplontic life cycle. Gametophyte N, gametogenesis syngamy, zygote 2N, meiosis, spores N, grows into gametophyte. Haplontic life cycle. 2. On the other extreme is the type wherein the diploid sporophyte is the dominant photosynthetic independent phase of the plant. The gametophyte phase is represented by the single to few celled haploid gametophyte. The kind of life cycle is termed as diplontic. An alga fucus species represents this pattern, figure 3.7b. In addition, all seed-bearing plants, that is gymnosperms and angiosperms, follow this pattern with some variations, wherein the gametophytic phase is few to multi-celled. Sporophyte, meiosis, gametophyte, gametogenesis, zygote, syngamy, zygote, sporophyte, diplontic life cycle. 3. Bryophytes and pteridophytes interestingly exhibit an intermediate condition, haploidiploidic. Both phases are multicellular. However, they differ in their dominant phases. Haploidiploidic life cycle, gametophyte N, gametogenesis syngamy, zygote 2N, sporophyte 2N, meiosis, spores, haplodiploidic life cycle. A dominant independent photosynthetic thalloid or erect phase is represented by a haploid gametophyte and it alternates with a short-lived multicellular sporophyte totally or partially dependent on the gametophyte for its anchorage and nutrition. All bryophytes represent this pattern. The diploid sporophyte is represented by a dominant independent photosynthetic vascular plant body. It alternates with multicellular saprophytic or autotrophic independent but short-lived haploid pteridophyte. Such a pattern is known as haplodiploidic life cycle. All pteridophytes exhibit this pattern. The diploid sporophyte is represented by dominant independent photosynthetic vascular plant body. It alternates with multicellular saprophytic or autotrophic independent but short-lived haploid gametophyte. Such a pattern is known as haplodiploidic life cycle. All pteridophytes exhibit haplodiploidic life cycle. Interestingly, while most algal genera are haplontic, some of them such as Ectocarpus, Polysiphonia, Kelps are haplodiploidic. Fucus and alga is diplontic. Summary. Plant kingdom includes algae, bryophytes, pteridophytes, gymnosperms and angiosperms. Algae are chlorophyll-bearing, simple thalloid autotrophic and largely aquatic organisms. Depending on the type of pigment processed and the type of stored food, algae are classified into 3 classes, namely Chlorophyceae, Phaeophyceae and Rhodophyceae. Algae usually reproduce vegetatively by fragmentation, asexually by formation of different types of spores, and sexually by formation of gametes, which may show isogamy, mesogamy, or oogamy. Bryophytes are plants which can live in soil but are dependent on water for sexual reproduction. Their plant body is more differentiated than that of algae. It is thallus-like and prostate or erect and attached to substratum by rhizoids. They possess root-like, leaf-like, and stem-like structures. The bryophytes are divided into liverworts and mosses. The plant body of liverworts is thalloid and dorsiventral, whereas mosses have upright, slender axis bearing spirally arranged leaves. The main plant body of a bryophyte is gamete-producing and is called a gametophyte. The main plant body of a bryophyte is gamete-producing and is called a gametophyte. It bears a male sex organs called antheridia and female sex organs called archegonia. The male and female gametes fuse to form zygote, which produces a multicellular body called a sporophyte. The male and female gametes fuse to form zygote, which produces a multicellular body called a sporophyte. It produces haploid spores. The spores germinate to form gametophytes. In pteridophytes, the main plant body is a sporophyte, which is differentiated into two root, stem, and leaves. These organs possess well-differentiated vascular tissues. The sporophytes bear sporangia, which produces spores. The spores germinate to form gametophytes, which require cool, damp places to grow. The gametophytes bear male and female sex organs called antheridia and archegonia, respectively. Water is required for transfer of male gametes to archegonium, where zygote is formed after fertilization. The zygote produces a sporophyte. The gymnosperms are plants in which ovules are not enclosed by any ovary wall. After fertilization, the seeds remain exposed, and therefore these plants are called naked seeded plants. The gymnosperms produce microspores and megaspores, which are produced in microsporangia, and megasporangia are born on sporophylls. The sporophylls, microsporophylls, and megasporophylls are arranged spirally on axis to form male and female cones, respectively. The pollen grain germinates in pollen tube, releases the male gamete into the ovule, where it fuses with the egg cell in archegonia. Following fertilization, the zygote develops in the embryo and the ovule into seeds. In angiosperms, the male sex organs, stamen, and female sex organs, pistil, are born in the flower. Each stamen consists of a filament and an anther. The anther produces pollen grains male gametophyte after meiosis. The pistil consists of an ovary enclosing one to many ovules. Within the ovule is a female gametophyte or embryo sac, which contains the egg cell. The pollen tube enters the embryo sac where two male gametes are discharged. One male gamete fuses with egg cell, syngamy, and other fuses with diploid secondary nucleus, triple fusion. This phenomenon of two fusions is called double fertilization and is unique to angiosperms. The angiosperms are divided into two classes, dicotyledons and monocotyledons. During the life cycle of any sexually reproducing plant, there is alternation of generation between gamete-producing haploid gametophyte and spore-producing diploid sporophyte. However, different plant groups as well as individuals may show different patterns of life cycle, haploid, diploid, or intermediate.